Buffaloes and cattle are very similar, yet different, bovine rumen animals. This study, directed by Lwin, focuses on ruminal fermentation and microbial ecology and how each differ between the two species. Throughout this 16-week trial, the cattle and buffaloes were fed napier grass and concentrates at a 1:1 ratio, and water and mineral blocks were supplied ad libitum. Rumen fluid samples were collected orally at 0, 3 and 6 hours after feeding, and pH was measured immediately at each interval. Ammonia levels were analysed using the Indo-Fennell method, and the methane production could be estimated using the molar proportion of short-chain fatty acids, as in genomic DNA extracted from the rumen fluid was analysed and used to create clone libraries of F. succinogene, the major ruminal cellulotic bacterium. The DNA samples collected through the trial were pooled in order to create DNA templates for both species. The rumen fermentation parameters, pH, ruminal short-chain fatty acids, ammonia nitrate levels and methane production can be attained using samples of short-chain fatty acids, as reflected in this table of fermentation profiles shown here. As indicated, many of the differences between the two species were not significant, particularly in relation to the time after feeding and the microbial fermentation factors of each species. Results show that rumen pH of buffaloes remained stable regardless of feeding, unlike cattle where the pH dropped significantly at 6 hours after feeding. The ammonia nitrate concentration clearly decreased after 6 hours, however this was not found to be statistically significant. Buffaloes held constant levels at 0 and 3 hours with a slight decrease at the 6-hour mark. The total of short-chain fatty acids was higher in cattle than in buffaloes, but every sampling time and the difference between was not significant. The proportion of acetate constantly decreased at 3 and 6 hours after feed in cattle, and in buffaloes the proportion was slightly higher at the 3-hour mark. The N-butyrate levels were similar in both species, with a slight decrease at the 3-hour mark and an increase at 6 hours. Throughout this study, Lwin and his team also measured the diversity of F. succinogene strains in the rumen and how the presence varied between the two species. The results reflected some differences between cattle and buffaloes, with many of ruminal cellulotic bacterium that were represented higher in cattle seemed to be those represented lower in buffaloes, and vice versa. The paper showed there is only a small amount of significant variation between cattle and buffalo in relation to the rumen fermentation parameters, which in turn reflects ruminal fermentation and microbial ecology differences. The buffalo's rumen microbes proved to be slightly more powerful than that of cattle, which is really reflective of their uses today, and may be evidence of their adaptations to modern day lifestyles.